Hello everybody, today we're going to do a 6 marker where it's talking about ultrafiltration and tissue fluid. So pause the video here, try this question out and continue on to see how I answer it. Right, this sort of question is dreaded by many people. It's because there's so much to think about and so much to process in your brain, right? Not only is it asking you about ultrafiltration, but also tissue fluid. So that kind of means like we need to think, what is it? What is ultrafiltration? What is tissue fluid? What are the similarities? What are the differences? It's just a bit long, isn't it? So the way I would do this is, of course, read the question first. It's not much. There isn't anything really to read. And the way I would go about it is just rephrase the question. So it's very simple. It's just like similarities and differences. But I would rephrase it into a bunch of questions. Because there's two different processes, I would ask myself, okay, what molecules do they have in common? Where is the location of these things occurring? What are the pressures like? What's actually happening? Is there reabsorption? What is there? What membranes are involved? Then I ask myself, like, what happens after the process or before the process or something, and like that. So in that way, I've got a bunch of questions that I'm thinking of actively while, I might be annotating around the question or the page about what on earth these two processes are, right? In the exam, of course, there's a bunch of topics where it's just thrown at you, opposed to like an end of topic test, where you just, yeah, it's going to be on plant responses, that's fine, that's it. But in the actual exam, you can have one question on like biotechnology and the next one on transport animals, right? So that's why you need to reduce the friction and the resistance between you not like thinking efficiently. And the way you can do that is, number one, ask yourself, what topic is this? Number two, just write, around the page what that thing is. So when you revise like ultrafiltration, and like you finish that memorization process and stuff, talk it out. Talk as if you've got a friend next to you and you're explaining this process to them, or just do it in person, really. And by doing that, it really, really does allow you to, in the exam, hear yourself back. It happened to me so many times in my A-level exams. I literally heard my voice. Like, yeah, there's that internal voice, whatever, but I heard the conversations I was having to the wall when I was describing the processes. Think out loud. Of course, you can't do that literally. I mean, think out loud in your head. But, So here, let's actually talk about it. So you always want to write out your answer in a methodical and logical approach. So if there's similarities and differences, just put 2 headings. Simple. Then, of course, now I'm assuming you guys know about ultrafiltration and tissue fluid. So from that, and from like answering the questions that I told you previously of what I would be thinking, right, I can start to make answers out of it. So let's talk about similarities here. Small molecules are filtered from the blood. They both, that's literally what happens in both of them. Very obvious. Both processes occur in capillaries. Well, of course, that's the location. So that's why it's really important to state all the obvious points, right? Like, yeah, it's in the capillaries, but some students actually forget to write that. They always get really, let's say, laser focused or like even tunnel vision into like, okay, I need to say all the nuances of each process and stuff like that. No, darling, take a step back. Take a step back. Just look at it for how it is. Next points are like large molecules, for example, proteins, high hydrostatic pressure. You always want to talk about pressure here. They both involve basement membranes. Just state all the obvious points, right? But then when it comes to differences, you kind of need to think a bit more, what's the word? A bit more, really. So for differences, we've got filtrate enters the Bowman's capsule and then PCT in the kidney, but tissue fluid, like, baths the cells, right? That point is kind of like the steps, right? The steps that occur in each process. So they would normally be the differences, whereas similarities are just like the things that are happening or are within that process, right? So molecules or the pressure or there's this membrane, there's that membrane, stuff like that. Next different point is that The molecules are not reabsorbed by capillaries from urine in the kidney, but molecules that are not reabsorbed from tissue fluid will form lymph. Again, it's like a step-by-step process, which has caused the comparison between both the processes, right? So in ultrafiltration, the molecules aren't reabsorbed by the capillaries from the urine, stuff like that. But then with tissue fluid, actually, the molecules that are not reabsorbed, they form the lymphatic, like they go into the lymphatic system essentially. So that was a lot, right? But it's very simple. You just need to know what the processes are, and then streamline your approach of it by asking yourself high value questions like what are the molecules involved, what membranes are involved, what are the pressures, what's happening or not happening, stuff like that. Okay, so this sort of question I would 100% add to my notes as like good recap really, and it's like free content notes as well if you think about it. A 2-in-1 special, I like to say. So that is the end of today's episode. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.